Move on to our next speaker, Ron van Bree.
Hello, everyone. I think the mic works, yes. So thank you for the opportunity for me to share my work here today. This is some work I did at the start of my PhD, which is on, believe it or not, combining mechanistic crop growth models with machine learning in order to improve their interpretability, as well as make sure the solutions they learn are in line with our understanding of how crops develop. The type of models that I'm interested in are these thermal time models, actually, that are quite simple, but like they form a core component usually of these biophysical crop growth models, and determine how the phenological progression of crops, which determines how susceptible crops are to environmental stressors often. So, and despite being quite a core component, they can show quite some structural discrepancies between models, which can have a large impact on simulated yields, for example. So the choice of model usually introduces quite a strong bias in your results. So we were interested to see whether we could use a more data-driven approach to learn something about the structure of these crop growth models. And at the time, the most interesting data set that we had on crop phenology actually was on cherry tree flowering, which also use these thermal time models and also have the same kind of problems. So we chose to focus on that. And yeah, so there are these works that show that using whatever machine learning model algorithm, they can achieve better prediction ability for these flowering events. But we were more interested in using the structure provided by these biophysical models to retain that interpretability. So to very briefly summarize how these models work, they generally consist of two phases, which each are modeled using these thermal time models. A chilling phase in which a sustained period of cold temperatures needs to be endured, followed by a forcing phase with warmer temperatures, which both need to accumulate to some threshold, after which flowering occurs. And both use their own temperature response functions to determine how hourly temperature levels contribute towards phenological development. And mainly in this first phase, there are quite some model discrepancies. So you really have to, yeah, there are all sorts of model candidates that are very distinct, and usually you really have to select and calibrate your model per environment, which can also have quite some impact on how these models behave. So how we dealt with that was quite simple. We just substituted this component that showed this discrepancy with a machine learning model, in this case a simple multi-layer perceptron, because we have this high dimensional hourly temperature input, and then our model outputs the contribution towards phenological development as captured by your biophysical model, let's say. We then approximated this biophysical model in a using a piecewise differentiable approximation, so we could compute gradients through our model with respect to both the machine learning parameters, model parameters, as well as the biophysical model parameters, and then we use these gradients to optimize our model. And some details, but the data set that we used was a data set on cherry tree flowering in Japan, Switzerland, and South Korea. It was very, quite a sizable data set compiled by the George Mason University Department of Statistics as part of a competition, actually. And we paired this data with temperature data as well as the tree species distribution, which you can see on the map. And especially in Japan, there's a very diverse range of climates in which the same tree species actually grows, and this is also reflected in the flowering dates. So in the south of Japan, the flowering really starts around February, whereas in the north this can be somewhere in June. So really the same tree species is very well represented in a very diverse range of climates. We chose to use 3 different evaluation strategies, and also included 3 popular biophysical models as a baseline, as well as a sort of naive deep learning LSTM baseline as well. In our first setting, we allowed the models to really choose or be fit using location-specific parameters. But in the second and third setting, we really asked ourselves the question, like, to what extent do you really need to fit these models per location, and can we try to instead fit these models per tree species? And then in the third setting, we try to evaluate these parameters in held-out locations. So what we observed is that each of the models was quite well able to predict these flowering dates, and surprisingly, despite the constraints imposed on our machine learning model, or maybe because of the constraints, the model, the hybrid model, if you will, showed better generalization ability here. But what is more interesting, I think, is that when we try to fit these models per tree species, we saw that the biophysical models just were not expressive enough to fit to the data in such diverse climates, whereas the hybrid model was able to learn a temperature response function for which it was possible to find such a set of parameters that was or worked reasonably well in a more diverse more diverse settings, and even when evaluating these on held-out locations, they seem to work relatively well. But then on to the maybe main question of why we had this model structure to have this interpretability. And we then inspected what did our model actually learn, and does it actually make sense? The brief answer to that is not quite. So we here show 3 temperature response curves as learned by our model for 3 different weight initializations in Japan. And what we see is that the initialization of the weights actually did matter for what kind of shape we end up with. So our model was kind of under constraint, and we have some equifinality issues going on. And the second thing that we observed was that the range, the temperature range at which these phenological contribution actually occurs is much wider than any of these biophysical models that we display on the bottom. From that we conclude that the function that we actually learned might not be quite biophysically plausible, let's say. So despite generalizing well, like we were able to predict these flowering dates quite well, while retaining the interpretability of our biophysical model. We were able to learn a model representation that for which it was able to learn parameters that seemed to work or that worked in a larger variety of climates. But when using the constraints imposed on our model to inspect what was going on. We did see some implausible behavior. Future work might be able to investigate whether using more regularization techniques we could try to further constrain or under constraint model to enforce biophysically plausible behavior while still retaining the same generalization abilities. Thank you very much.
All right, thank you, Ron. Does anyone have one question for Ron?
Yes, so many questions.
Thanks very much. Really interesting study. I was wondering, I didn't quite get what the genetic structure of your data was. So you referred to different species.
Yeah, it's really species level.
So, okay.
It's actually it's compared to many of the talks here. It's a very coarse or like very high level, abstract level of.
Okay, because that might have some relevance to your model performance
Right,
as well, right?
yeah,
Because the trait that you looked at is probably pretty heritable, pretty strongly sort of genetically determined. But yeah, we can.
Yeah, no. So we really just focus on really the species level, but it might be interesting to try and extend this type of work to see if you could go to a lower level even.
Okay, so how many species were these? Sorry, just.
So we focus on 3 species.
3. All right. Okay.
Yeah.
Thank you.
Yeah.
All right, thank you, Ron.
Please.